Okay, thanks for responding. So, I'll start this session. So, today is the seventh problem solving session for the course Environmental Chemistry and Microbiology. So, for the past six sessions, we have been discussing topics from environmental chemistry. From today onwards, we'll start the microbiology part. So, today's session is all about introduction to microbiology, how what is the history of microbiology, how the uh, field in particular evolved over time, how uh, different discoveries were made, and then what are the classification system in biology and particularly in microbiology, and some basic information. So this will be very this will be like an introduction to microbiology and history of microbiology. So I have certain questions prepared in this session which are related to your assignment questions. So I hope all of you will answer to these questions. And if you have any doubt in any of the questions, please ask. If you have any doubt in any of the topics in general, uh, then also please let me know. I'll try to help you as much as possible. Okay. Uh, okay. Okay, so I'll start with the questions. I will request all of you to write the answers in the chat box and then we'll have a discussion if your answer is correct or not. So, starting with the first question, which of the following famous person was first to find out that microorganisms are responsible for wound infection and develop the method of antiseptic surgery? Options are option A, Robert Hooke, option B, Paul Ehrlich, option C, Joseph Lister, and option D, Louis Pasteur. Which of the following famous person was the first to find out that microorganisms are responsible for wound infection and develop the method of antiseptic surgery? Robert Hooke, Paul Ehrlich, Joseph Lister, and Louis Pasteur. All of you write your answers in the chat box. I have three answers in the chat box. All of them are option C. What about others? Okay. Anyone else wants to answer? Who developed the method of antiseptic surgery? Okay. So four answers are there. All of them are option C. So let's see the correct answer. The correct answer is option C, Joseph Lister. So all of you were correct. So Joseph Lister basically developed the antiseptic surgery procedures that we nowadays follow very frequently. So whenever we get a cut, we apply some antiseptic liquid, either Dettol or some similar solutions, right? And the concept behind using all those uh, antiseptics was basically developed by Joseph Lister. So he found the way to prevent infections in the wounds during and after surgery. So he found out that during surgery or after surgery, patients were developing infections because of certain bacterial growth over the wound. Okay, or during surgery, there's some uh, if someone is doing surgery and they have not sterilized the equipments that they are using for surgery, those equipments can be responsible for transmitting various bacteria and microorganisms. Okay, and those bacteria can grow on the open wound and it can develop some infections. So he realized that there are certain microorganisms that were responsible for development of such infections or uh, such sepsis, septic infections. Okay, so then what he did was he used carbolic acid as an antiseptic at that time, and he used the carbolic acid during the surgery to clean the wound area and all, and that actually prevented the development of infection, and that was the concept he applied, that we need to use certain agents that can kill the microbial growth and that can prevent the microbial growth from happening in the open wound, so that we don't develop infections. So during the time of war, uh, people were dying not because of a disease that they had, but because of the post surgical infections. So there was a dying need to prevent those infections, because if someone had some certain uh, medical problem, they were not the actual reason for their death. The reason for the people dying was actually the procedures that were applied to treat the treat particular condition, and those procedures were leading to certain kind of infections. So he was the first to apply the science of germ theory to surgery. So uh, germ theory is basically states that a particular microorganism can cause a particular disease, and if you isolate that particular microorganism and grow it, it should grow separately, and then if you put that microorganism in a healthy individual, the disease will develop. So that is the germ theory, and he applied the germ theory here in the uh, infection and post surgical infection area, and then he found out that uh, importance of usage of antiseptic during the surgery or on an open wound. So the techniques that we use in today's day and age for antiseptic has been, like, the foundation has been built by Joseph Lister. Okay. The other options that were there: Robert Hooke, Paul Ehrlich, and Louis Pasteur. So Robert Hooke was the one who first observed a cell under a microscope, not a microbial cell, a normal cell. And then Louis Pasteur has multiple accomplishments in the field of science. He's the one who has developed or explained the process of fermentation. So process of fermentation has lived a long time in our uh, in this world, but uh, to find out the reason behind fermentation, that microbes are the ones who are called uh, responsible for the fermentation. So we all eat bread. Bread is basically a fermented dough. That fermentation happens because of a microorganism called yeast. Similarly, we all eat dosa or some form of fermented snack. In India, there are many fermented food items that are available. So all the fermentation process are due to presence of one or the other bacteria or fungus. Okay. Then he also disproved the spontaneous generation theory, and then he is the developer of the procedure of pasteurization, which is a sterilization technique. You all must be well aware with the term pasteurization. All the milk packets that we get, you might have seen that it is written pasteurized milk. So it's a sterilization process at a very high temperature. The milk is heated for a very brief time so that all the microorganisms are killed in that uh, milk, and there is no so it can be stored for a longer time. So it's a preservation process for a sterilization process for preserving it for a longer time. Then Paul Ehrlich was the one who gave the effective treatment of syphilis, and he developed the drug for syphilis, which was called salvarsan. Okay. The next question is: the defining characteristic of living beings are Options are they reproduce, they move in response to external stimuli, they grow and repair their own tissues, and all of the above. What are the defining characteristics of living beings? Uh, Write your answers in the chat box. Okay, I have a question in the chat box. What is the meaning of disproved spontaneous generation? So, spontaneous generation was a theory which stated that the living matter, like living matter, came from the non living matter. So, non living matter gave rise to the living beings. But Louis Pasteur actually performed an experiment which disproved this theory. So basically, he showed that a living organism can only arise from a living organism. It cannot arise just from the non-living matter that is present. So that was Louis Pasteur's work. So he had performed an experiment in a swan flask. You can read about it in detail. So he had in I think in two in like in one round bottom flask, he had in one case he had the tube attached to the round bottom flask, and I don't exactly remember the experiment, but then in the round bottom flask he had the tube attached, and in one case he closed the tube, and in one other case the tube was open. So in the for case where the tube was open, he showed that there were growth of certain microorganisms because uh, things could pass through the flask or something like that. So I'm not very sure of the experiment. You can read about the Louis Pasteur's experiment, swan neck flask experiment. If you search for it, you will find. But spontaneous generation basically means that it was a theory which stated that living things came from the non-living matter, or non-living matter gave rise to the living matter. But he disproved that. He said that living things can only arise from living cells. One living cell can give rise to more living cells. Okay. uh, okay, we were in on second question. Defining characteristics of living beings are, I can see most of you have answered option D, all of the above. That is the correct answer. So, defining characteristics of living beings, all of the characteristics that are mentioned here the uh, are the defining characteristics for living beings. So they reproduce, they move in response to external stimuli, and they can grow and repair their own tissues. So there are certain criteria, you can say, which define a particular particle, a living or a non-living uh, object. So if we want to call something living, it should metabolize. First of all, metabolism means the cell should be able to take nutrient from the environment and transform it into biomass and energy, and it should excrete out the waste into the environment itself. So the cycle should go on. The metabolic cycle should be present in every living cell. So if a cell is living, there should be a metabolism pathway. So for us, we eat food, then we convert the food into biomass and energy. We grow because of eating the food, and we gain energy because of eating the food, and the output, the waste, is excreted out, which goes back to the environment. And then the food that we are eating is also, we are getting it from the environment. So there's a metabolic cycle. So for every living cell there is a metabolic cycle. So if there is metabolism present, that is one characteristic of living cells. Then second characteristic is reproduction. So if the cell can divide and reproduce, it should be called a living cell. Third characteristic is differentiation. Differentiation Cells can adapt to a new environmental condition, such as changing into spores to survive in hostile conditions. So, a living cell will always be able to adapt to a new environmental conditions. Okay, be it a larger more evolved organism, or be it a single celled bacteria. Okay, even if it is a single celled bacteria, they also have certain mechanisms. If you suddenly change the environmental condition of that bacteria, it has certain mechanisms to adapt to that new environment slowly, and then start growing there. And that is exactly how we have such antibiotic resistance and conditions like that. So I am hoping you all are aware of the antibiotic resistance. So when we take antibiotics, antibiotics are there to kill the bacteria, but there are certain bacteria, what happens is the environment around them has antibiotic now. So there are certain bacteria who are trying to adjust to that new environment, and now they have developed certain genes, like they have evolved, and they have gained certain mechanisms to resist that antibiotic, so that they don't die in that environment but they can live. So that is one very important characteristic of living cell, that they can differentiate and they can adapt to a new environmental condition. Okay, next characteristic is communication. So every living cell has a process of communication. It need not be as simple as we are communicating by talking to each other, but every cell has a method of communicating. Even inside your body, the cells are communicating with each other by one means or the other. Either they are releasing some chemicals, or they are signaling some kind of changes around their environment, so that the next cell next to it uh, uh, gets the message, and they can then respond accordingly. So even bacteria, they release certain chemicals, which another bacteria which is there in the surrounding, it uh, recognizes, and then it says, okay, this bacteria wants me to respond in such a way, then I'll do so. So there are ways of communication and in every organism, be it a plant, animal, or a unicellular organism. And then comes movement. Movement, again, every organism, we walk, so the movement is very apparent. But if you see, plants can't walk, but they also move. How do they move? They move in response to light. Okay, there are certain organisms that can move in response to certain chemicals, certain pheromones. So there are movement. If there is a movement, it's a characteristic of a living cell. Then comes evolution. Evolution is one very important characteristic of living cell. The cells that can modify themselves to adapt to new condition, resulting into new species over long period of time. So differentiation and evolution has this particular difference, that evolution is a slow process, whereas differentiation is a immediate adaptation in some certain hostile condition. Like uh, if suddenly the environment around me becomes very cold, my body will have certain responses which can make me adapt to that situation temporarily. But if I am forced to live in that condition for ages, then slowly our body will start evolving according to the condition. Like the theory of evolution that has happened in the on our planet basically happened because of the change in the environment itself. So the way the planet's environment started changing, slowly the organisms also evolved to adapt to that changing condition, to the new condition. Okay, is this clear to everybody? uh, Mobini wants me to show the question number 1 and 2. I can show it to you. So this was the question 2, the defining characteristics of living beings. And this was the question 1, who developed the who was responsible for finding out microorganisms can cause wound infection and developed the method of antiseptic surgery. Okay.
Thank you, ma'am.
Yeah. uh, yes, Swastika, you can ask the question. Can you ask the question at the end of the class, if that's okay? Then I'll finish, otherwise we'll break the flow. Okay. So I'll quickly finish the topics. Today's questions are anyway theoretical, so it won't take longer time. Then we can discuss. So moving to the third question. Does anyone have any question regarding the previous two questions? Any doubt or anything you want to ask regarding the characteristics or regarding the history that we discussed? So moving to the third question. If you have any question, you can write it in the chat box. I'll read it. Question number 3. Who explained the cause and effect relationship between a pathogen and a disease? Who explained the cause and effect relationship between a pathogen and a disease? Options are Edward Jenner, Vinogradsky, Van Leeuwenhoek, and Robert Koch. Who explained the cause and effect relationship between a pathogen and a disease? Write your answers in the chat box. Yeah, I can see people answering option D. So all of you have answered option D, Robert Koch. That is the correct answer. So the cause and effect relationship between pathogen and disease, this was explained by Robert Koch, and this was explained by uh, the Koch postulates. He gave four postulates. Uh, and if you remember from the first question, I told you about the germ theory. Uh, so the germ theory is actually comes and like it comes under the Koch's postulates, where it the postulates go like the microorganism must be found in abundance in all organisms suffering from a disease but should not be found in healthy organisms. So I'll just give one example and I'll explain it with that example. So if one individual is suffering from malaria, okay, so there should be one organism, one microorganism that should be present in that particular individual, but it should be absent in the person who does not have malaria. In this case, that person will be called as healthy organism. Okay, so that microorganism cause malaria causing microorganism that should be present in the person who has malaria, but it should not be present in the healthy organism. That is the first postulate. Second was the microorganism must be isolated from the diseased organism and grown in pure culture. So we should be able to isolate that microorganism from the person who has malaria and grown it in pure culture, and I can grow it in pure culture. Then third is cultured microorganism should cause disease when introduced into a healthy organism. So once I grow it in pure culture, if I now take that microorganism and put it in the person who does not have malaria, he should develop malaria. So that is how the uh, transmission of disease, or that is how the germ theory, where one organism can grow and that organism can be, like should be, uh, like we should be able to isolate that organism, grow it in pure culture, and then again put that organism in some other individual. Okay, so that is how the disease progression takes place. And the fourth postulate is microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. Now, initially, I took the microorganism which was causing malaria. I grew it in pure culture from the diseased individual. I took it out and then I grew it in pure culture. And then now, if I take that microorganism and put it in a healthy individual, that healthy individual should develop malaria. And if I now again take the uh, microorganism from the individual who has recently developed malaria and compare it with the previously isolated microorganism, it should be identical, like both should be identical, uh, concluding that the causative agent is indeed this particular microorganism. So if it is TB, we know that Mycobacterium tuberculosis is causing TB. If it is malaria, there's it's a parasitic disease. So Plasmodium falciparum is causing this disease. So we should know, like whenever we get some bacterial or viral infection, we get it tested in our blood, right? That means that microorganism should be detectable in our blood, should be cultured. So how do they detect? Mostly they have some or the other marker to check whether you have that particular microorganism. And then in some cases they actually isolate that microorganism by taking, like, putting a swab of your blood sample on agar plate so that that microorganism grow, and they can actually isolate that microorganism and check whether you have that particular microorganism or it is something else. So the so all those techniques that we use now, that we use today, are actually developed way back by these uh, people. Okay, so Robert Koch is one of the very important person in microbiology who has developed this theory, where he showed, now when we are reading it, it sounds very redundant that this should this like this is something we already know, but there was a time when this was also not known, and at that time he actually gave this postulate, and then people got to know. And based on these postulates, Joseph Lister understood that during like on a wound, there are certain microorganisms that are growing that are causing infection. If he takes that microorganism and put it in some other wound which which does not have infection previously, it will also develop infection. So you see how these things are connected. I hope all of you understood this. So apart from this, the options state Edward Jenner. So Edward Jenner was the one who developed the smallpox vaccine. So what he did was he took a sample from a patient who had cowpox, which was similar to smallpox but a less lethal version. Smallpox was very serious, but cowpox was not that serious. So he actually took the lesions that were there on the patient uh, when they developed cowpox. So pox is basically you get uh, rashes kind of things on your skin, right? I am hoping you all are familiar with chickenpox, because that is something we all had in our generation. So, what Edward Jenner did was he took the lesion from a patient who had cowpox, and then he gave that particular uh, mixture with the things that came out from the lesion. He then injected that particular mixture in a patient who did not have smallpox initially, and then observed that whether he developed smallpox or not, and he did not develop smallpox later. So that's how Edward Jenner got the idea of small of the vaccine development. Like if we actually inject a small amount of uh, causative agent, like in case recent everyone has gotten vaccinated for COVID, right? So I'll give that example. So if we now know that if we give a modified version of this virus to the patients who have not yet developed COVID, their body will now recognize that particle as, okay, this is something we need to fight against. And then when you actually encounter a COVID virus, coronavirus, what happens is your body recognizes, and your body, because it already has the immune system developed against that because of the vaccination, it will immediately fight and prevent your body to develop the disease. Okay, so that is the concept behind vaccine, and that concept was developed by Edward Jenner. Next was Vinogradsky. He pioneered the cycle of life concept. So he developed a Vinogradsky column where uh, it was actually distributed, like he created a mini ecosystem. I think we will discuss about it in some of the next sessions. Then comes Leeuwenhoek. Leeuwenhoek is known as father of microbiology. He was the first one to observe a bacterial cell under a microscope. So he developed a microscope on, like, on his own, and then he observed the bacterial cell. So Robert Hooke developed a cell for the first observed a cell for a first time, and Leeuwenhoek observed a bacterial cell for a first time. Okay, so these are all history of microbiology, and if you are working in this field, you should be aware of these things. Okay, because this is how the field evolved. Okay, this is just some major milestones in microbiology. You can go through it. This is from the lecture slides only. Okay, moving on to the next question. Question number fourth. Prokaryotes can be found in the following two domains. Options are bacteria and protista. Option B, archaea and eukarya. Option C, bacteria and archaea. And option D, eukarya and monera. Prokaryotes can be found in the following two domains: bacteria and protista, archaea and eukarya, bacteria and archaea, eukarya and monera. All of you write your answers in the chat box. Okay, I see most of you have answered. Option C, bacteria and archaea. That is the correct answer. So prokaryotes are so there are two classes, prokaryotes and eukaryotes. Living organisms are divided into two types, prokaryotes and eukaryotes. Prokaryotes are unicellular, whereas eukaryotes can be unicellular or multicellular. Under prokaryotes, we have archaebacteria or archaea and eubacteria, or commonly we call it bacteria. So archaea and bacteria are under prokaryotes, whereas eukaryotes, they have unicellular and multicellular organisms. Unicellular organisms include algae, protozoa, and fungi. So there are some fungi which are unicellular, like yeast, and there are some fungi that are multicellular, like the mushroom you eat, that is also a fungi, and that is a multicellular fungi. Okay, and then comes Plantae and Animalia. The whole kingdom of Animalia and Plantae comes under the multicellular eukaryotes. The difference between prokaryotes and eukaryotes, there are multiple characteristics which are different for both prokaryotes and eukaryotes, starting with the size of the cell. For prokaryotes, since they are unicellular organisms, they are very small in size, 0.2 to 2 micron in diameter typically, whereas eukaryotes, they are bigger cells which has 10 to 100 micron in which are 10 to 100 micron in diameter. Nucleus, this is a very important difference between prokaryotes and eukaryotes. Prokaryotes actually lack the membrane bound organelles. Okay, so there are no membrane enclosures inside the prokaryotes. That is the major difference between a prokaryotic cell and a eukaryotic cell. Eukaryotic cell will have internal membrane bound organelles. So nucleus is absent in prokaryote, whereas nucleus, because nucleus is also a membrane bound organelle, it is the organelle in which the DNA or the genetic material is present. Okay, so eukaryotes have a nucleus, and it contains the genetic material, whereas in prokaryotes there is no nucleus as such present, but the genetic material is actually localized to one region. So everything is floating in the cytoplasm like in one single pool, but the genetic material, it's not just floating around throughout the cell, it is actually localized to one region. The difference is it is not surrounded by any membrane, whereas for eukaryotic cell it is actually surrounded by a lipid membrane. Okay, so that is the difference between the prokaryotic cell and a eukaryotic cell. The region where the prokaryotic genetic material is localized, that region is called nucleoid. Okay, then comes chromosomes. There is only single circular chromosome present in the prokaryote. The genetic material is usually circular, and there is only single uh, circular DNA present in the prokaryotic cells. Whereas eukaryotic cells, they have multiple linear chromosomes with histones. So in eukaryotic cell, the genetic material is organized into more complex structures. So it is more structurally organized, you can say. So the DNA is uh, folded, and since it's a bigger cell, you can imagine the genetic material is also larger. So there are more base pairs present in the DNA, and the DNA length will be longer. So the DNA is more compact, and they are more structurally arranged. They are arranged into like the DNA thread, it is actually wrapped around proteins called histones, and then those are again compressed to form chromosomes. And we have multiple such chromosomes, not one but multiple. So humans have 23 pairs of chromosomes. Okay, then comes membrane enclosed organelles. I already told you prokaryotes lack any membrane enclosures, whereas eukaryotes have multiple organelles. So you must have heard about mitochondria and Golgi apparatus and endoplasmic reticulums. Everything is present in eukaryotic cell but not in a prokaryotic cell. Then comes flagella, consists of two protein building blocks in prokaryotes, but eukaryotes have complex, consists of multiple microtubules. Then glycocalyx, it is present as a capsule or slime layer in prokaryotes, whereas in eukaryotes it is present in some cells that lack a cell wall. In prokaryotes, cell wall is usually present and chemically complex. Typical bacterial cell wall includes peptidoglycan. We will discuss this later. And in eukaryotes, mostly it is absent, and when it is present, it is chemically simple. So, eukaryote animal cells does not have a cell wall, but plant cells do have a cell wall, and it is not chemically that complex. Then plasma membrane, there are no carbohydrates and generally lacks sterols in prokaryotes, but eukaryotic plasma membrane has sterols and carbohydrates embedded in the lipid membrane. Cytoplasm, I told you there is no cytoplasmic streaming because there is no compartmentalization, but in eukaryotes there is compartmentalization, so there is cytoskeleton present, there is cytoplasmic streaming. Then ribosomes, the smaller size ribosomes are present in prokaryotes, whereas in eukaryotes, both larger and smaller uh, ribosome is present. Cell division occurs mostly by binary fission in prokaryotic cell, but in eukaryotic cell it occurs by mitosis and meiosis. So the division of prokaryotic cell, or the reproduction of prokaryotic cell, is mostly asexual by binary fission. So it just one cell splits into two, two cells splits into four, and go move and so on. But in eukaryotic cell it is more complex because eukaryotic cells have the capability to undergo sexual reproduction system, where both the process of division, which is mitosis, and meiosis, where the division is not it's not 1 to 2, 2 to 4 like that. Okay, so it involves both mitosis and meiosis. Okay. So the difference between prokaryotes and eukaryotes is clear to everybody, I hope. If you have any doubt, please let me know. Moving on to the next question. Match the following organisms to appropriate kingdom. So there's one column which shows the organism, and the other column shows the kingdom. Okay, the organisms are Escherichia coli, Entamoeba coli, diatoms, Loria formosa, and Penicillium chrysogenum. And the kingdoms are algae, animalia, fungi, bacteria, and protozoa. Okay, so you have to match the organism with the kingdom. Write your answers in the chat box. I already have one answer in the chat box. Right. All of you try to answer. Okay. I see four answers. All of them are option D. What about others? Anyone else wants to answer? Okay, I see option D in the chat box. Let's see. So, starting with Escherichia coli, it is a bacteria. So, 1D. There's only one option with 1D. Entamoeba coli, this is a protozoa. Okay, so 2E. Then diatoms, it's a class of algae. So 3A. Then comes Loria formosa. It's actually a type of mite, I think yellow mite. So 4B. So it's a animalia. Uh, so it is one type of insect, you can say. And then Penicillium chrysogenum, it's a fungus. It's a very common fungus. So 5C. Okay, so option D is the correct answer. So all of you who answered option D, very good. So Escherichia coli, it is also known as E. coli. It is a gram-negative facultative anaerobic rod-shaped coliform bacteria of the genus Escherichia that is commonly found in the lower intestine of the warm-blooded organism. So it is present in our gut, and it is also a very common bacteria that is used in the lab as model organism. So any kind of DNA preparation or protein purification, if you want to do it in bacterial system, E. coli is the choice. Everybody, it's a very, very commonly used organism. Okay, and it is easy to grow. That doubles in every 20 minutes. So in like 16 hours, you can have a lot of cells, and you can easily, like if you want to overexpress some particular protein, you can easily put the gene of your interest, and you can ask the bacteria to make the protein for you. So that is the concept. Then comes Entamoeba coli, which is a protozoa. It's one of many non-pathogenic protozoa found in humans. It is transmitted through fecal or oral contact, and the mature cysts can be found in contaminated water. Typically, these protozoa inhabit the large intestinal tract and can be identified in diagnostic stool specimens. Then diatoms are the major group of algae, as I told you, specifically microalgae found in oceans, waterways, and soils of the world. Loria formosa, commonly known as yellow mite or the citrus yellow mite, it is a species of acariform mite. They are a subfamily of Tidini, of the family TDD. Then comes Penicillium chrysogenum, which is a species of fungus in the genus Penicillium. It is common in temperature and in temperate and subtropical regions and can be found on salted food products, but it is mostly found in indoor environments, especially in damp or water damaged buildings. So the fungal growth that you see on your damp walls, sometimes those are mostly penicillium growth. So if you have any questions regarding this, you can write it in your chat box or you can unmute and ask. Are there any questions? Okay, then moving on to the next question, question number 6. Overall shape of a bacteria is determined by which of the following? Options are cell wall, cytoplasm, nucleoid, and cell surface membrane. Overall shape of a bacteria cell is determined by which of the following? Cell wall, cytoplasm, nucleoid, or cell surface membrane. Write your answers in the chat box. All of you write the answers so that I'll at least get to know if you are understanding or not, or if you know the answer or not. And if you don't know the answer, and even after my explanation, if you have any doubt, please ask. This session is for your doubt clearing. So I see 5 of you have answered option A, cell wall. What about others? Anyone else wants to answer? There are 15 people in this uh, meeting. I think I should get more than 5 answers. Anyone else? There's nothing to think about. All these are theoretical questions. So overall shape of the bacteria is determined by which of the following? You all are saying option A, cell wall. That is the correct answer. So cell wall is the outermost covering of the bacteria, most of the bacterial cells, and that is the rigid structure that defines the shape. So basically, a bacterial cell or a prokaryotic cell is prokaryotic cell has the outermost layer, which is the cell wall, then comes the plasma membrane, and then comes the cytoplasm, in which all the genetic material, the nucleoid where the DNA is present and the ribosomes, everything is floating in the cytoplasm. Okay, so you have the outermost layer, the cell wall, then the membrane, plasma membrane or cell membrane, and then comes the cytoplasm inside the membrane. Okay, the cell wall is composed of uh, uh, chemical or a material called peptidoglycan. So by the name itself, you can see it has peptide and glycan. So peptidoglycan. So bacterial cell wall is the characteristic of bacterial cell wall is presence of peptidoglycan. Okay. So peptidoglycan, as the name suggests, there are glycans, N-acetyl glucosamine and N-acetyl muramic acid. So there are alternating units of this NAG and NAM. So these are abbreviated as NAG and NAM. So NAM and NAG units. There are alternating units of that. So you have one NAM, one NAG, and then again NAM, and so on. And from these glycans, there are peptide, a small tetrapeptide. You can see the composition here. The small tetrapeptide is attached, and then pentaglycine is cross-linked. So total 9 amino acids are attached to this glycan. So that's why it is called a peptidoglycan. Okay. Now there are two types of bacterial category, we can say, based on the thickness of peptidoglycan in the cell wall. So there is one that is called gram positive bacteria and one that is called gram negative bacteria. The gram positive bacteria has a thick layer of peptidoglycan in the cell wall, and the gram negative bacteria has a thin layer of peptidoglycan in their cell wall. So this is the plasma membrane, and after plasma membrane, outside of the plasma membrane, we have the cell wall. So you can see for gram positive, there's a thick layer of peptidoglycan cell wall, And for gram negative, there's a thin layer of peptidoglycan, and outside of that there is a thin outer lipid membrane also. There's an additional membrane that is present, lipid membrane, that is present in the gram negative bacteria. Now, why it is named as gram positive or gram negative, it is actually given by this person who developed a staining procedure, and the staining procedure is also called gram positive or gram negative, gram staining basically. So in gram staining, what we do is, first we take, like, if let's say I don't know whether the bacteria is gram positive or gram negative, so for that I'll take the bacterial cell and make a smear on the slide. I'll put, first we'll add crystal violet, and uh, we'll add few drops of crystal violet. So what will happen, the gram positive bacteria, since there's a thick peptidoglycan layer, the crystal violet will bind to this cell wall, and it will stain the cells purple. For the gram negative bacteria also, it will stain the cells purple, but after crystal violet we add fixing solution, and then the mordant, basically the iodine solution, and then we use the decolorizing agent, that is ethanol. So when we decolorize it, because the peptidoglycan layer is thin in case of gram negative bacteria, the crystal violet is easily removed from the cells. But for gram positive bacteria, the since the peptidoglycan layer is thick, the crystal violet does not leave the bacteria so easily, with even after decolorization. And then we stain it, counter stain it with another dye called safranin, which is purple in which is pink in color. So since the gram positive bacteria already has the crystal violet and they are violet in color, they can't now take the pink color, or it is not that clearly visible, pink being the lighter color. But in gram negative bacteria, after decolorization, the crystal violet has left the cells, so these cells can get easily stained by safranin, which is pink in color, and you can easily distinguish the gram positive violet, dark violet colored cells, uh, as opposed to the gram negative, which are pink colored cells under the microscope. So that's how the name gram positive and gram negative came. Okay, is this clear to everybody? Any doubt in this? Any questions? Moving on to the next question. What is the genome size in million bases of yeast? What is the genome size in million base of yeast? Options are 12.1 million, 4.6 million, 137 million, or 97 million. What is the genome size of yeast? So yeast is a unicellular fungus, as I've already mentioned. So think of it in that way. What is the genome size of yeast cell? Okay, I see two answers in the chat box. What about others? Anyone else wants to answer? Okay, two more people have answered. Okay, all of you are answering option A, 12.1 million. That is the correct answer. That is the correct answer. The genome size of yeast is 12.1 million bases. So this is just a list of different organisms. So you can see how varied the genome sizes and the estimated number of genes are there in all the organisms. So starting with humans, we have a 3.2 billion bases. The genome size is 3.2 billion, which is a huge genome size. If you think of it, and it has estimated 30 to 40,000 genes. Laboratory mouse, it has a smaller genome size as compared to us, 2.6 billion, but number of genes are almost similar to humans. That's why we use these mouse as the model organism to test certain compounds to do clinical trials and all. Okay, like before clinical trials, preclinical trials are done on these lab mice. And then comes mustard wheat, which is Arabidopsis thaliana, which is a plant, and it is actually an interesting model organism in the among the plants, because the genome size, if you see, it is very small. It is, I think, the smallest genome among plants. Okay, so that's why, since it is small, it is just 100 million as opposed to 3.2 billion. The sequencing of Athaliana was done very early, and the number of estimated genes are 25,000. And then comes roundworm, C. elegans, another very important model organism that we use in labs. Generally, C. elegans have a translucent type of body, so it is very easy to track if you want to see the localization of some protein, where it is present inside the cell. Such kind of studies are very easily done in C. elegans. And the genome size is 97 million. The number of genes is 19,000. Then comes fruit fly or Drosophila melanogaster, again a very important model organism which is used in, especially in developmental biology, studies related to developmental biology. There are certain genes in Drosophila which are very similar to the human developmental genes, and it serves as a very good model organism. Then comes yeast. It is used in many different areas in labs as well as in food industries, for fermentation processes. I told you bread is also fermented by yeast. The scientific name is Saccharomyces cerevisiae. The genome size is 12.1 million, and the number of genes are only 6,000. Then comes bacteria. The common example is E. coli, and E. coli has a 4.6 million bases genome size and 3,200 genes. It is, I told you, it's a very common, the most common organism that you will see in any biology lab. And then amongst viruses, HIV has a very small genome, only 9,700 bases and only 9 genes. Whereas coronavirus, the genome size is much larger than HIV. HIV has only 9 genes. Do you know why? Because viruses do not reproduce or do not grow uh, on themselves. Like, they can only grow and they can only reproduce when they are inside the host cell. So coronavirus also, it couldn't have reproduced on its own if humans were not present. So it survived for so long because, and it is still surviving, uh, and the reason it is surviving and it survived, the pandemic survived for so long, because there were many hosts in which it could multiply easily, and that's how it was increasing in number. If there were no hosts present, and that's hence why the lockdown was uh, like it started, so that we can minimize the host contact of this virus, so that we can control the growth of the virus. So viruses can't grow outside the host organisms, and thus HIV, with only 9 genes, it can survive very happily, because it is anyway going to use the rest of the genes from the host itself. So it uses host's machinery. So if a virus is infecting my body, the virus is not going to use anything of its own. It does not have most of the genes. It will use my genes and my proteins and make its own copies. So that is the smart mechanism of viruses, and that's how such pandemic like situations occur. Okay, so anyone has any doubt, you can ask. If you have any question, let me know. Okay. Moving to the next question, question number 8. Flow of genetic material in microbial cell usually takes place from, options are RNA through DNA to protein, option B DNA through RNA to protein, option C protein through RNA to DNA, and option D none of the above. Flow of genetic material in microbial cells usually take place from RNA, DNA, protein, DNA, RNA, protein, protein, RNA, DNA, or none of these. What would be the answer? Write your answers in the chat box quickly. What would be the answer? Okay, I have one answer in the chat box. All of you write the answers quickly. All of these are theoretical questions. There's nothing to calculate or solve. Okay, I see 4 answers in the chat box. What about others? Anyone else wants to answer? Okay, you all are saying option B. 5 answers are there. All of them are option B, DNA through RNA to protein. That is the correct answer. So, the genetic material in the microbial cell is DNA. The information from the DNA it is passed into RNA and then into protein. So, this is the central dogma of biology, where from the DNA is transcribed into RNA. The process is called transcription, and then the RNA is translated into protein. The process is called translation, and that's how the information, the genetic information, or the codes that are there in the DNA, it is converted into protein through RNA. Okay. So information is stored, processed, and replicated. So first DNA is replicated, like more copies of DNA are formed, and then it is uh, transcribed, as I mentioned, by process transcription, and then the next process, translation, occurs, where the RNA serves as a template for producing specific protein. Okay. Any doubt in this? Let me know if you have any doubt. Okay. Moving on to the next question. Property of a codon for always coding a specific amino acid is called. Property of the codon for always coding a specific amino acid is called. Options are degenerate, non-overlapping, non-ambiguous, or none of these. The property of a codon for always coding a specific amino acid. Option A, degenerate, option B, non-overlapping, option C, non-ambiguous, and option D, none of these. What would be the answer? Write the answers in the chat box. Only 2 answers. Okay. I see most of you have answered option C, non-ambiguous. That is the correct answer. So, a single codon always codes for the same amino acid. This describes the non-ambiguous nature of genetic codon. So, codons are in triplet form. So, these are the properties of codon. The code is in triplet. The code is degenerate, non overlapping, comma-less, unambiguous or non ambiguous, and universal. So, there are four bases present in the DNA: A, T, G, and C. In case of RNA, the T becomes U. Okay, so AUGC or ATGC. Now, there are four bases and the codon is in triplet. So that means we have 4 into 4 into 4 probability of having, like, the number of combinations of that we can get, triplet combinations that we can get out of 4 bases would be 64 codons. Okay, out of those 64 codons, we have 61 codons that code for amino acid and 3 codons that are called stop codon, which tells us where to end the translation process. Okay, so from these 61 codons, now how many amino acids do we have? We have only 20 amino acids and we have 61 codon. So every codon codes for one amino acid, that is the unambiguous nature or non-ambiguous nature of the codon. But there are certain amino acids which are coded by more than one codon. These codons are not overlapping with each other. There are no comma between these codons. These are continuous triplets. So if the codon is ATG or GAG or anything GTA, so it is comma-less. Okay, and these are universal. So ATG is coding for methionine, so it will code for methionine in my body also, in your body also, in a plant, in a bacterial cell, in every cell. ATG will code for methionine irrespective of the organism. So it is universal. Okay, any questions in this? The next question is, the genetic code is degenerate code because option A, there are 64 codons for 20 amino acids. Option B, one codon has many amino acids. Option C, each codon codes only for one amino acid. Option D, more than one codon codes for single amino acid. The genetic code is called degenerate code because there are 64 codons for 20 amino acid. One codon has many amino acid. Each codon codes for only one amino acid, or more than one codon codes for single amino acid. What would be the answer? All of you write the answers in the chat box. Why the genetic code is called degenerate? I have 3 answers in the chat box. Anyone else? I just explained the properties of genetic code. I thought this should be answered by more people. Only 3 of you have answered. No one else wants to answer? So 3 of you have answered option D. More than one codon codes for single amino acid. That is the correct answer. So I told you there are 61 codons, not 64. 61 codons coding for 20 amino acids. The rest of the 3 codons are stop codons. One codon codes only for one amino acid, and that property is called non-ambiguous or unambiguous. One codon does not have many amino acid. That is a wrong statement. And more than one codon codes for single amino acid, because obviously you have only 20 amino acids and 61 codons. So obviously more than one codon has to code for one amino acid, and that property is called degenerate property of genetic code. So you can see here, sorry, you can see here the codons. So UUU and UUC, both of them code for phenylalanine. Similarly, you can see CCU, CCC, CCA, and CCG. All four of them codes for proline. So there are four codons. Each codon is coding for one amino acid, but four of them, all of them are coding. So irrespective of, like in my body, if CCU is present or CCC is present, both of them will code for proline. Okay. Similarly, there are codons which all four of them are coding for arginine. There are codons, all two of them are coding for serine. So these are the genetic codes which are present. So, and you can see three of them are stop codon: UAA, UAG, and UGA. Okay, so there are 64 combinations of three-letter codon. I explained three-letter nucleotide sequence. I explained how. Of these 64, 61 are used to code 20 amino acids, and 3 are stop signals. Although each codon is specific for only one amino acid, genetic code is described as degenerate or redundant because a single amino acid may be coded by more than one codon. Okay, I hope that's clear to everyone. Anybody has any doubt? Since only 3 of you have answered, I hope this is clear to others as well. Let me know if you have any doubt. Okay, moving to the next question, question number 11. Studies of similarities and differences among the genomes of multiple organisms is carried out in. Options are proteomics, functional genomics, structural genomics, and comparative genomics. Study of similarities and differences among the genomes of multiple organisms is carried out in proteomics, functional genomics, structural genomics, or comparative genomics. What would be the answer? Write your answers in the chat box. Okay. Again, I can see 3 answers. 3 of them are option D. What about others? Option D, that is comparative genomics. So 4 of you are saying option D. Let's see the correct answer. The correct answer is comparative genomics. So this is a study of the similarities and differences. So basically it's a comparison of genomes from different organisms. Okay, by the name itself you can understand it is comparative genomics. Proteomics is actually the study of all the protein present in a particular organism. So proteome is the set of proteins produced in an organism, system, or biological context, and large scale study of these proteomes is called proteomics. Functional gene, like the term genomics comes from the study of genes. So proteomics comes from study of proteins. So under genomics we have functional genomics, structural genomics, comparative genomics. So functional genomics means we are focusing on the dynamic expression of gene products in a specific context, like uh, at specific developmental stage or during a disease or some particular condition. Like I am changing the condition and seeing what is happening to the function of that gene. Basically function of that gene is to produce some product, some protein. So what is happening to the expression level? So that is functional genomics, expression of the gene, not the protein itself. For if we are studying the protein, then it will become functional proteomics. Okay, here we are solely talking about the expression of the gene. So under certain diseased condition, maybe one gene is expressed more or less than the normal condition. So that is functional genomics. Structural genomics, it refers to the high throughput three-dimensional structural determination and analysis of biological macromolecules. So like functional genomics, we are Checking the function, in structural genomics, we are focusing on the structural, three-dimensional structure. Okay, that's clear to everybody. Moving on to the next question, question number 12. Which of the following may be used as RNA as its genome? Which of the following may use RNA as its genome? Sorry. Which of the following may use RNA as its genome? Options are bacterium, archaeon, virus, and eukaryote. Which can use RNA as its genome? Write your answers in the chat box quickly. These are very straightforward questions. All of you should write the answers quickly. Okay, good. Most of you are answering option C, virus. That is the correct answer. So, most of the organisms, almost all the organisms have DNA as the genetic material or the genome. But there are certain viruses which can have RNA as the genetic material. And when it infects the host organism, it actually converts the RNA into DNA and then uh, replicates and then again makes it back into RNA and then packages into the virus particle. So the most common example of a RNA virus I can give is the coronavirus itself. It was a RNA virus, and if you remember or if you know about the testing procedure that was followed uh, for detection of COVID, it was basically a RT-PCR method. So RT stands for reverse transcription or reverse transcriptase. So the RNA was converted back to DNA. So I told you DNA to RNA process is called transcription. So the reverse is called reverse transcription. So RNA was converted back to DNA, and then that DNA was undergoing the PCR cycles so that we can detect whether you have that particular viral infection or not. Okay, that virus is present in your sample or not. So there are the only one here that may have RNA genome is a virus. So virus is the correct answer. Moving on to the next question. Anybody has any questions regarding this, regarding the discussed things till now? The next question is, what is the type of extremophile that thrives in the area with very little water where it is really dry? Options are halophile, thermophile, xerophile, and radioresistant. What is the type of extremophile that thrives in areas with very little water? Halophile, thermophile, xerophile, or radioresistant? What would be the correct answer? Write your answers in the chat box. Okay, I can see 5 answers already. All of them are option C, xerophile. What about others? Anyone else wants to answer? Okay, I see most of you are answering option C, xerophile. That is the correct answer. So xerophiles are the organisms that can grow and reproduce in conditions with very low availability of water. Halophiles are the ones which can tolerate high salt concentration. Thermophiles are the one which can tolerate high temperatures, and radio resistant, as the name suggests, it can survive in high amount of radiations. It can resist high radiations. Okay. What is the type of extremophile that lives in very salty environment such as Great Salt Lake? I just gave you the answer. You can still answer. What would be the type of extremophile that lives in salty environments? I just gave you the answer, so all of you should know this. I see only two answers. Yeah. So all of you are saying option A, halophile. That is the correct answer. So halophiles can survive in high salt environments. So the oceans, the Great Salt Lake, which has a very high salt uh, uh, presence of salt concentration uh, in such environment, in such water bodies, the bacteria that are present, they are generally halophiles. They can tolerate that amount of salt concentration. Then comes thermophiles. So all of you must be aware of hot water springs, where the water is naturally hot due to the presence of certain minerals uh, underground. So, and in such hot water springs, also there are certain bacteria that can survive, and such uh, bacteria, they are called thermophiles. Xerophiles I've already explained. Radiophile also I've explained. Okay, so you should know examples of these halophile, thermophile, xerophile. So thermophile, there's a popular example that we generally give. It comes from the enzyme that is commonly used for PCR cycles. So we use Taq polymerase, so Thermus aquaticus. It is actually a thermophile, and that is why the polymerase isolated from that organism is able to work at higher temperatures. Like, it does not get degraded. There are other organisms also which are nowadays uh, PFU polymerase is also used. So that is another bacteria that is also isolated from one of the bacteria, which is a thermophile. Similarly, for halophiles, for radiophiles, there are different examples given. You should look into it and you should remember at least one example. Okay. Halophiles are the group of organisms with the ability to prosper in hypersaline conditions. In this kind of extreme environment, the salt concentrations are in excess of seawater and can reach up to 20 to 30% NaCl, which is a very high concentration, just for your information. Halophiles require high salt concentrations to thrive in these unusual habitats. Moving on to the next question. Anybody has any doubt, you can ask. The next question is, binomial name of a microbe is composed of. The binomial name of the microbe is composed of. Options are its kingdom and genus name. Option B, its genus name and order name. Option C, its family and class name. Option D, its genus and species name. What would be the answer? I can already see answers in the chat box. What about others? All of you write the answer in the chat box. Binomial name of microbe is composed of. Okay, I see four answers in the chat box. All of them are option D. Its genus name and species name. That is the correct answer. So the names that we have discussed so far, like Escherichia coli. So Escherichia is the genus and coli is the species. Similarly, what else we discussed? Penicillium chrysogenum. So Penicillium is the genus, chrysogenum is the species. So the nomenclature of the bacteria, it refers to naming and bacteria and naming of bacteria and other organisms. They are named according to the binomial system, which was introduced by Carl Linnaeus. This means that a bacterium has a name which is composed of a genus name that tells you which genus it belongs to, and the species epithet, which together with the genus name, that becomes the unique name for the bacterium. So there's only one bacterium called Escherichia coli. Okay. Similarly, there's only one fungi that is called Penicillium chrysogenum. So as an example, Moraxella bovis, where the genus indicates the bacterium belongs to the genus Moraxella, and the species epithet indicates that the bacterium has been isolated from cattle. So it is isolated from a bovine species. That's why the name bovis is added. So it depends. For Escherichia coli or E. coli, coli comes from the colon. I told you it is present in our gut, it is present in our colon. So it comes from that. And there are certain other, like sometimes the species name comes from the shape of the bacteria, or the genus name comes from the shape of the bacteria. So it depends. Okay. And it is always written in italics. When we are writing it uh, by hand, we can't differentiate between normal and italics font. So in that case, what we do, we make small underline between underline that particular genus and species name. So if I'm writing Moraxella bovis, I will just put a line under the genus and the species name. Okay, and when I'm typing something, I'm always supposed to write the genus and the species name in italics font. Okay, to differentiate that, okay, this is the gene, this is a organism name, as opposed to all the other words. If you want to spot, it is easier to spot, and this is just a defined way of writing the names. So if even if I'm writing Homo sapiens, where Homo is the genus and the sapiens is the species, I have to write it in italics. So Homo sapiens is written as italics. It is the scientific name for humans. Okay. Next question is question number 16. The gram negative bacteria that causes stomach ulcer is, options are Helicobacter pylori, Bacillus anthracis, Clostridium botulinum, and Neisseria meningitidis. The gram negative bacteria that causes stomach ulcers. Option A, Helicobacter pylori. Option B, Bacillus anthracis. Option C, Clostridium botulinum. And option D, Neisseria meningitidis. What would be the correct answer? Write your answers in the chat box quickly. Which bacteria causes stomach ulcers? Okay, I see 4 answers in the chat box. All of them are option A, Helicobacter pylori. Anyone else wants to answer? The correct answer is option A, Helicobacter pylori. It is a gram-negative bacteria and it causes stomach ulcers. The other options, Bacillus anthracis, it causes anthrax. It's a commonly uh, it commonly affects the domestic and wild animals around the world. People can get sick with anthrax if they come in contact with the infected animals or contaminated animal products. Okay, and that, to prevent anthrax, we generally do the sterilization of animal products like milk. Okay, then comes Clostridium botulinum. It is a bacteria that produces dangerous toxin, that is botulinum toxin, under low oxygen condition, and this toxin can uh, is one of the most lethal substances known, and it can block the nerve function and can lead to respiratory and muscular paralysis. But the interesting thing about this botulinum toxin is, if you have heard of Botox, I'm pretty sure most of you must be aware of the term Botox, and people, generally the celebrities, they get Botox done on the skin, so to prevent wrinkles, right? To prevent looking old. So how does that act? That Botox is actually this botulinum toxin. Short form is Botox. And how does it work? So if you see here, it is written that it blocks the nerve function and can lead to respiratory and muscular paralysis. So it is a very lethal substance, but in very small dose, it can, if that very small dose is injected directly into the affected areas where wrinkling is more prone, in such areas your muscle becomes paralyzed, and thus it cannot grow further and droop down, and that's how wrinkles are basically a muscle movement, and muscle is drooping downwards. So if you paralyze the muscle there, then there won't be any wrinkles present. So that is the, like, the negative part of the toxin was used for a cosmetic procedure later. Then comes Neisseria meningitidis. By the name itself, it causes, you can understand, it causes meningitis or meningococcal disease, which is commonly known as brain fever. It affects the central nervous system. Okay. Anybody has any questions or any doubts? Okay, the next question is, who isolated Streptococcus pneumonia for the first time? Options are Robert Koch, Edward Jenner, Anthony von Leeuwenhoek, and Louis Pasteur and George Sternberg. Who isolated Streptococcus pneumonia for the first time? Robert Koch, Edward Jenner, Leeuwenhoek, Louis Pasteur, and George Sternberg. Write your answers in the chat box. Okay. Option D, I can see most of you have answered. Option D is Louis Pasteur and George Sternberg. That is the correct answer. So they isolated the Streptococcus pneumonia for the first time, and they actually isolated it uh, in their individual labs. And later they got to know that both of them have isolated simultaneously the same uh, bacteria, Streptococcus pneumonia. Okay. Next question is, newly discovered microscopic structure is hypothesized to be living organism. Which of the following lines of evidence would support the contention that this organism may be alive? Options are, it contains DNA, it is made up of single cell, it utilizes energy, and option D, all of these. Which of the following lines of evidence would actually support the contention that the organism may be alive? What would be the answer? Write your answers in the chat box. Okay, I see you are answering option D, all of these. That is the correct answer. We have already discussed the characteristics of a living cell, right? So this is same thing, just the question is framed in a different way. So since it's a microscopic structure, it should be made up of a single cell. It has to contain some genetic material, mostly it is DNA, and it should utilize energy. I told you metabolism is one of the characteristics of living cells, so this is something I have already discussed. The next question, a virus is considered to be non-living when it is outside the host, because this also we have discussed why a virus is uh,
Ma'am, could you please show us the previous slide?
yeah,
Thank you, ma'am.
yeah. Should I move on?
Yes, ma'am.
Okay. So a virus is considered to be non-living when it is outside the host, because options are, it cannot reproduce, it cannot utilize energy, it is mobile, and it is made up of a single cell. Which of the following option is the reason why virus is considered non-living? Write your answers in the chat box. I can already see three answers. All of them are option A. I've already discussed how virus function. So option A, it cannot reproduce. That is the correct answer. So four of you, five answers are there. So that is the correct answer. It cannot reproduce. So I have already told you how virus uh, acts. So it cannot reproduce on its own. It has to infect a host. It has to enter a host body where it will use the host's mechanism to reproduce and multiply and make more copies of itself, and then it will be passed to the other host, and then it will keep on dividing. Okay, so it cannot reproduce and it cannot utilize energy. So both A and B are actually correct. Okay, moving to the next question. Question number 20. What is Whittaker's classification based on? Options are mode of nutrition, cellular organization, presence or absence of nucleus, and option D, all of these. What is Whittaker's classification based on? Whittaker's classification is based on. Write your answers in the chat box. Options are mode of nutrition, cellular organization, presence or absence of nucleus, and option D is all of these. I can see three answers in the chat box already. All of them are option D, all of these. What about others? Anyone else wants to answer? Okay, option D, all of these is the correct answer. So, Whittaker's classification is based on multiple characteristics, uh, mode of nutrition, cellular organization, presence and absence of nucleus. So, Whittaker actually classified the organism into five kingdoms. So, these are the five kingdoms: Monera, Protista, Fungi, Plantae, and Animalia, amongst which I've already told you the cells are divided into two types, prokaryotic and eukaryotic, right? So the type of cell is the first characteristic he used. So he divided the prokaryotics from the eukaryotics. So he made the kingdom Monera, which consists all the prokaryotes, and the rest of the kingdoms, Protista, Fungi, Plantae, and Animalia, they contain the eukaryotic cells. Then it is also defined by the type of cell wall. So the non-cellulosic cell walls are present in the Monera criteria. In Protista, the cell wall is present in some and absent in some. Fungi, there is a cell wall present in all fungus cells, and it is made up of chitin. So chitinaceous, generally it is called chitinaceous cell wall. Then in plants also, I told you plant cells have cell wall and it is made up of cellulose. In animals, the cell wall is absent, there's only cell membrane. Nuclear envelope, I told you prokaryotes don't have the nuclear envelope, whereas eukaryotes have. So except Monera, all the other four kingdoms have the nuclear envelope. Cellular organization, now there are some cells which are unicellular, some cells which are multicellular, and some cells which are even organized into higher levels. So Monera and Protista are consists of unicellular fungus. They have both unicellular and multicellular types. So unicellular type of fungus is yeast cells, whereas multicellular type of fungus are multiple examples are there. Mushroom is one of the most common examples. Then plants and animals, they actually have a higher level of organization. So the become cells come together to make tissues, and then tissues come together to make organs, and in animals these organs come together to make organ systems. So like circulatory system or digestive system, these are all systems. So digestive system have multiple organs which come together to form the whole system. So we have stomach, we have intestine. So all these organs, they make the organ system, that is digestive system. There are tissues that make these organs, there are cells that make these tissues. So there are higher level of cellular organization in plantae and animalia. Then comes the mode of nutrition. We'll discuss this in detail in the next question. The next question is, photoheterotrophs are, options are, they get energy from light and feed on another organism. Option B, they get energy from light and are self feeding. Option C, derive energy from chemicals and are self feeding. Option D, derive energy from chemicals and feed on other organisms. What would be the correct answer? Write your answers in the chat box quickly. I can see 3 answers in the chat box. What about others? For what are photoheterotrophs? Okay, I can see all of you have answered option A. Get energy from light and feed on other organisms. That is the correct answer. Photoheterotrophs, they get energy from light and they feed on other organisms. So the mode of nutrition that we were talking about in the previous slide, the under Whitaker's classification. So based on the mode of nutrition, there are different types present. So all organisms, they can either feed on other organism or other organic carbon sources. So other organisms are basically organic carbon sources, or they can make their own food, like we say for plants. They can make their own food, they can photosynthesize, so they can directly use the CO2 present in the environment as the carbon source. So either they can use the organic carbon as the carbon source, or they can directly use the inorganic CO2 as the carbon source. If they are using CO2 as the carbon source, they come under autotrophs. If the organism is using organic carbon uh, as the carbon source, they are called heterotrophs. Okay, so this is the first level of division. Then comes the type of energy they are using. So if they are using light energy to use this carbon source, they are called photoautotrophs. Or if they are using light energy to use this organic carbon, they are called photoheterotrophs. And if they are using chemical energy, then it is chemoautotrophs or chemoheterotrophs. So in the question it was photo or heterotrophs. So photoheterotrophs, basically they are using light energy, they are gaining the energy from light, and they are feeding on other organism. Basically they are feeding on organic carbon. Okay, that's why they are called photoheterotrophs. Now there are further classifications present. So under photoautotrophs, if they are using H2O to reduce this CO2, then it is oxygenic photosynthesis. There's a presence of oxygen. So in plants, algae, they all the photosynthetic organisms commonly we study, these are oxygen. They are doing the photosynthesis in the presence of oxygen or in the presence of H2O. If they are not using H2O, if they are using some other sources, they that process is N-oxygenic photosynthesis, which is carried out by certain green and purple bacteria. So they use H2S instead of H2O to reduce the CO2. Then under chemoautotrophs, we have hydrogen, iron, uh, sulfur, nitrogen, and carbon monoxide oxidizing bacteria. So these bacteria can oxidize all these uh, compounds using the chemical energy. Then under photoheterotrophs, we have the green and purple non-sulfur bacteria, which can uh, use the light energy to break down the organic carbon. And chemoheterotrophs again are divided into two cases. If the terminal electron acceptor is oxygen, it is aerobic respiration, like we all are undergoing aerobic respiration. We breathe in oxygen and we use that oxygen for making energy. Okay, and then if the terminal electron acceptor is not oxygen, then in that case there can be two conditions. Either it is using organic compound, in which case we call it fermentation, like lactobacillus, it ferments the milk, or it ferments other products by using organic compound, by using the compound that is present. Like if it is fermenting some batter, it is using the components of that batter to do the process. It is not. And generally, if you recall from your household kitchen and all, so you can see any fermentation, whenever we are keeping a batter to ferment, we keep a lid on it. The reason to keep a lid on it is because we want to cut the oxygen supply in the batter, because the fermentation is an oxygenic procedure, or we don't want it is the terminal electron acceptor is not oxygen. If the oxygen is present, then there will be no fermentation process. The process will always be towards the aerobic respiration. Okay. And if the oxygen is not present and the fermenting bacteria are present, they will undergo fermentation. And if the inorganic compounds are used, then it is called anaerobic respiration. Example is clostridium undergoes anaerobic respiration. Is this clear? Anybody has any doubt in this? The next question is, which of the following nitrogenous base is purine? So options are adenine, thymine, cytosine, and uracil. All four bases are given. So tell me which of this is not a sorry, which of this is a purine? Write your answers in the chat box quickly. Which of the following is a purine? Adenine, thymine, cytosine, and uracil. Yes, two of you have answered option A. Okay, more people are answering. So option A, I see all of you are answering option A. Option A, adenine. That is the correct answer. So there are two classes in the nitrogenous bases, purine and pyrimidine. So pyrimidine has a one ring structure and purine has a double ring structure. So the basic structure defines the classes. So in purines you can see cytosine, thymine, and sorry, in pyrimidines you can see cytosine, thymine, and uracil are present. The only difference is in the side chain. The basic ring structure is similar in all three of them. And under purine we have adenine and guanine, where again you can see the side chain is different, the basic ring structure is same. Okay. The next and the last question is, which of the following differentiates a plant cell from an animal cell? Presence of ribosome, presence of cell wall, presence of nucleus, and none of the above. Which of the following differentiates a plant cell from animal cell? Presence of ribosome, cell wall, nucleus, and none of the above. We have already discussed this. I can see two people have already answered. Okay, more are answering. Option B, presence of cell wall. We have already discussed this. Plant cells have a cell wall made up of cellulose, whereas animal cells, they lack the cell wall. Okay. So these are the basic differences between plant and animal cells. So cell wall is absent in animal cells. Chloroplast is another difference. Chloroplast is present in plant cells, which have the pigment chlorophyll that gives the plants green color, and which is the pigment that is used that helps during photosynthesis, and that is absent in the animal cells because we don't photosynthesize. Then lysosomes are also generally absent in plant cells but present in animal cells. So these are the basic differences. Rest everything is almost present in both the plant and animal cells. Okay. So this was the last question of this session. Anybody has any doubt in this session? Uh, Somebody wanted to ask me a question. Swastika wanted to ask a question.
Yes, ma'am.
Yeah,
It was from Radioactive.
yeah, yeah. Ask.
Wait, Yes, the question was which of the following isotopes is used in the determination of age of dead plants and animals? I I marked 14 carbon, but the correct answer is 13 carbon. But I think it will be 14, right?
C14, generally carbon dating is done using C14 carbon. So it is showing wrong like the answer is given as C13. The answer
Ma'am,
is
in my case also the same thing happened. After evaluation I saw and the answer is showing C13.
Yes, ma'am. Same.
okay. I think they will change the answer. Like in the previous years, also, I have noticed sometimes their answer key has a wrong answer. So later they send an email saying that this particular answer was wrong. So it should be C14. So carbon dating is done using C14. So that is the correct answer. If they have marked C13, it's a team's error. I think they will correct it out. Don't worry. Okay. So they generally update the marks later. So at the end of all these sessions, you will get a mail, or you can also write a mail to the NPTEL team. There is a support team, right? So you can one of you can write email saying that this particular answer, although you all have marked it correctly, it is showing wrong. It should be C14. So your answer is correct. Don't worry. Okay. Anything
Okay, ma'am.
else? Yeah. Anyone else has any question regarding this session or any of the sessions? What was the exact question? Can you tell me the carbon thing?
Which of the following isotopes is used in the determination of the age of dead plants and animals?
Okay. Yeah, it should be C14. It should be C14. I think they have made a mistake. While making the answer key, they will update the answer. Don't worry. If you want, you can write a mail to the NPTEL support team. They can correct the answer, or just wait. They've generally did. I have seen in the previous years also, if they have made a mistake in the answer key, they generally correct it out. So don't worry. Okay. Uh, are there any other questions? Let me know. I'll wait. Mm, if there are no questions, then I think I can end the session. Everything is clear from today's session.
Yes, ma'am.
Okay. Okay, then thank you all for joining. I'll end the session here.
Thank you, ma'am.
Yeah, thank you. Bye bye.